All right, let's jump right in. Today, we're going to map out a huge and honestly super exciting territory, the world of next-generation sequencing, or NGS, data analysis. Think of this as a journey, taking us from a chaotic flood of raw data all the way to some really incredible biological insights.
So here's the thing. We can now sequence an entire genome in like a day, which is just mind-blowing, right? But that incredible power creates a totally new problem. What on earth do we do with all that data? It's just this mountain of raw information that on its own doesn't really mean anything.
To really get a sense of this data mountain, let's talk numbers. 3 billion. That's about how many DNA base pairs are in just one human genome. And the tricky part is, the sequencing machine doesn't just hand you a neat single file. Oh no. It spits out millions and millions of tiny, jumbled up pieces. We call them reads. And our job? It's like trying to solve the world's most complicated jigsaw puzzle.
So that's what we're here to do today. We're going to chart a course right through all that complexity. We're going to break down exactly how analysts take that chaotic mess of raw data and turn it into real, meaningful discoveries. Using a clear, step-by-step philosophy you'd find in places like NGS 101.
Okay, let's dive in. This is our starting point. No matter what biological question you're trying to answer later on, every single NGS analysis starts right here. It's all about taking that noisy, messy, raw stuff that comes off the machine and turning it into a clean, organized data set that we can actually use.
And I think this is so important to hear. You do not need to be some kind of programming wizard to get started in this. Seriously. The whole philosophy here is about making it accessible. The goal is to give biologists and researchers the power to answer their own questions, building up those skills from the ground up.
Okay, so this is what we call the universal workflow. It's basically the foundation for everything that comes next. Step 1 is quality control. I mean, it makes sense, right? If your raw data is junk, your results are going to be junk. Garbage in, garbage out. Then comes alignment. This is that giant jigsaw puzzle I mentioned. We're taking all those tiny reads and mapping them back to a reference genome to see where they fit. And last but not least, quantification. This is where we literally just count. We count how many reads landed on each gene, and boom, suddenly we have our first real biological signal, how active each gene is.
All right, so once we have that clean, quantified data ready to go, now the real fun begins. This is where we can start asking those big biological questions. So let's check out the first major destinations on our map.
So you can pretty much break down the whole world of NGS into 3 main fields, and each one asks a totally different kind of question. First, you've got genomics. That's asking what's actually written in the DNA. It's the blueprint, the permanent code. Then there's epigenetics. This asks, how is that code being regulated? Think of these as the light switches that turn genes on or off. And finally, transcriptomics. This one asks, okay, but which genes are active right now? It gives you a perfect snapshot of what a cell is doing in that exact moment.
But here's the thing. Biology isn't static, right? It's always moving, always changing. So we can go beyond these basic maps. We can start exploring dynamic systems and ask how all these different parts work together. And this is especially true when we zoom in, way in, to the level of a single cell. And honestly, this is one of the most exciting frontiers in biology right now. For so long, we studied tissues by just grinding them up. We were looking at a blend of thousands of cells all at once. It's kind of like listening to an entire orchestra playing every note at the same time. You just get noise. But now, with single-cell analysis, we can listen to each individual instrument. And the level of detail, the complexity we're finding, it's just incredible.
With something like single-cell RNA-seq, the questions we can ask get so much more detailed. Like, how do cells actually talk to each other? We can figure that out. We can literally watch a cell change and develop over time. We can even figure out which genes like to work together in teams, or find rare tumor cells hiding in a sample. It's a total game changer for understanding how life works.
So, you want to answer all these amazing questions, right? Well, it's not just about knowing the biology. To really explore this world, you need the right gear. You need a specific set of computational tools. Think of it as your analyst toolkit.
Because modern bioinformatics is really a mix of 2 different worlds. On one hand, you've got the science. You have to know your biology, your genetics. But on the other hand, you've got the tools. You need to be comfortable with high-performance computing, managing your software, and even a bit of programming. To really make progress, you absolutely need a foot in both camps.
But let me be clear again. The goal isn't to turn you into a master coder overnight. That's not the point. The point is to have a good map.
It's about knowing which tool to grab from your toolkit for which specific biological question. It's about being a smart, efficient, scientific explorer.
So that brings us to the horizon. Think about where we started. We had this massive data problem. What do we do with billions of DNA reads? And now look, we've mapped out this entire landscape of incredible biological questions that we can actually start to answer.
And this idea from raw reads to results really brings our whole journey full circle, doesn't it? The entire field is built on this one simple, powerful idea. You take raw, messy data, you follow a series of logical steps, and you transform it into a clear result that tells a compelling biological story. Okay, so let's do a quick recap of our map. We started with that universal workflow, getting our data from raw to clean. Then we explored the main territories: genomics, asking what's written; epigenetics, asking how is it controlled; and transcriptomics, asking what's happening now. And to top it off, we took a peek at the amazing frontier of single-cell biology, where we see how it all comes together.
And at the end of the day, that's what this is all about. Learning these skills gives you a map, but it's not just a guide to follow. It's an invitation to go out there, take all that raw data and turn it into brand new knowledge. So now that you have the map, what are you going to discover?